What if some of the most important life on this planet has always been too small for you to ever see, and yet has been shaping your world in ways you've never fully noticed? Right now, across your skin, in the quiet air around you, and deep within your body, countless living things are moving, exchanging, building, and transforming, not in sudden or dramatic ways, but in slow, continuous patterns that have been unfolding for billions of years. They have helped shape the air you breathe, the soil beneath your feet, even the rhythms inside your own body. They survive in places that seem impossible, endure changes that erase entire worlds, and continue in ways that are not always easy to imagine or immediately understand. These are bacteria, not a single kind of life, but an entire hidden world, ancient and ongoing, present in nearly every place life has ever taken hold. Tonight, we're going to move gently through that world, exploring how they began, where they exist, and what allows them to continue in so many different forms, one quiet fact at a time. There is nothing you need to remember. You can simply listen, and if you drift away along the way, the story will continue without you. So settle in, let the day soften, and we'll begin with the oldest life this planet has ever known. Bacteria are the oldest known living organisms on this Earth, with fossils reaching back approximately 3.5 billion years into the deep past. Think, for a moment, what that number truly holds inside it. 3.5 billion years ago, this planet was something unrecognizable. No forests, no oceans filled with fish, no soft grasses bending in any wind. There were only warm, shallow seas and bare, cooling rock, and the slow, patient chemistry of a world still learning what it meant to be alive. And yet, already, there was life. Small, invisible, uncelebrated life, but life nonetheless. Those first bacteria did not know they were beginning something. They simply were. They simply continued. And here, billions of years later, their descendants are still here. In the soil beneath your home, in the quiet interior of your own body, in the clouds drifting above you as you lie still and breathe. Bacteria have been here longer than anything else that has ever lived. They are the elders of all living things. The first bacteria may have appeared when the Earth was as little as 400 to 500 million years old. Chemical signatures pressed into ancient rock suggest that life may have stirred as far back as 4.1 billion years ago, though the earliest confirmed fossil evidence dates to around 3.5 billion years. The Earth itself is approximately 4.54 billion years old. What this means, in its quietest and most extraordinary sense, is that life did not wait very long at all. By cosmic reckoning, it arrived almost immediately, like something that had always been waiting just off stage, patient and ready. Scientists find this deeply striking. A young, violent planet, still being struck by meteors, still settling into itself, still finding the shape of its continents. And already, in its warm and chemical waters, something was learning to grow, to divide, to continue. Life did not need a finished world. It began in the middle of becoming. Cyanobacteria were the first organisms on this planet to produce oxygen through photosynthesis, and in doing so, they slowly, over billions of years, filled the sky with the very air we now breathe without thinking. This process has a name, the Great Oxidation Event, and it occurred roughly 2.4 billion years ago, long before any creature existed that could have witnessed it. Imagine that, a world whose atmosphere was being quietly, steadily, invisibly transformed by tiny beings too small to see, working not with intent but simply with the blind and beautiful chemistry of their own existence. Every breath you are taking right now in this soft and sleepy moment carries the legacy of those ancient cyanobacteria. The oxygen filling your lungs as you sink deeper into rest was, in a very real sense, a gift given by beings who lived before the first fish, before the first leaf, before the first footprint ever pressed into any mud. Stromatolites, layered, rocky structures built over immense stretches of time by ancient cyanobacteria, still exist today in places like Shark Bay in Western Australia, rising gently from the shallow saltwater like quiet monuments to the oldest life on Earth. They are not dramatic to look at. They are low and dark and slow and easy to overlook. But if you were to sit beside them at the edge of that warm Australian water, you would be sitting beside something almost incomprehensibly old. The stromatolites in Shark Bay are living descendants of structures that were already ancient when the first complex animals were just beginning to emerge. They have been building themselves layer by quiet layer for billions of years. They do not hurry. They do not need to. They have already outlasted nearly everything that has ever tried to hurry on this earth. Some scientists have reported reviving bacterial spores from salt crystals estimated to be 250 million years old, though this claim remains genuinely contested, with many microbiologists cautioning that the samples may have been contaminated by modern bacteria during extraction. Whether or not that particular study holds, the story it points toward is real. Bacteria can enter a state of dormancy so profound, so complete, so perfectly still, that they become almost indistinguishable from the minerals around them. They stop growing, they stop dividing, they wait. They are not dead, and they are not quite alive in the way we usually mean. They exist in a kind of suspension, a biological pause, like a breath held at the very bottom of an exhale. The question of how long that pause can last is one that scientists are still very gently trying to answer. Bacteria have survived all 5 of Earth's major mass extinction events, the same catastrophic collapses that erased the dinosaurs, that silenced vast swaths of ocean life, that reset the story of life on this planet and forced it to begin again. Each of those events was a kind of ending. The sky went dark, or the oceans acidified, or the temperatures swung far beyond what most life could endure. And yet, through every one of those endings, bacteria remained. They persisted in the soil, in the deep rock, in the sediment at the bottom of seas that were turning hostile. They did not conquer those disasters. They simply outlasted them, quietly, without drama, in the way that very small and very ancient things sometimes do. In this way, bacteria are the great survivors of the living world. Every catastrophe has passed over them like weather. The last universal common ancestor of all life on Earth, the one living thing from which every bacterium, every plant, every animal, every human being is descended, was likely a single-celled organism closely resembling modern bacteria. Scientists call it LUCA, and it existed somewhere between 3.5 and 4 billion years ago, probably in the warm, mineral-rich waters near a hydrothermal vent deep in a primordial ocean. LUCA was not the first living thing, but it was the ancestor that all surviving life traces back to. Everything that has ever lived and left descendants shares, in some deep and distant way, a common origin with those first bacteria drifting in a young and volcanic sea. You are related, in the most ancient sense of that word, to every bacterium that has ever existed. The family tree of life begins in the smallest possible place. Ancient bacteria helped form the very first soils on this planet, breaking down bare, lifeless rock into something soft and layered and rich enough to one day hold a root. Before bacteria went to work on it, the surface of the early Earth was stone, hard, cold, unbroken, offering nothing to anything that might want to grow. But bacteria are patient chemists. They produce acids. They break mineral bonds. They weave themselves into the cracks of rock, and slowly, over geological stretches of time, they unmake what was hard and remake it into something living. The topsoil beneath a garden, the rich, dark earth of a forest floor, the soft loam that a seed presses itself into on a spring morning. All of it carries at its origin the slow and ancient work of bacteria. They did not know they were making the world habitable. They were simply alive. Bacterial life existed in Earth's oceans for nearly 2 billion years before eukaryotes, cells with a true nucleus, appeared. Though bacteria were not entirely alone during that long span, the archaea, a separate and deeply ancient domain of life, likely emerged around the same time and shared those primordial seas alongside them. Together, bacteria and archaea held the world between them, invisible and tireless, cycling nutrients, breathing gases, transforming the chemistry of the oceans and the sky. For 2 billion years, life on Earth was entirely microscopic, entirely invisible, entirely silent in any way we could perceive. There were no eyes to see, no ears to hear, no minds to wonder. There was only chemistry, endlessly folding in on itself, growing more complex, more varied, more resilient with every passing era. Life was patient with itself in those long-ago oceans. It had all the time the universe could offer. Some of the iron ore deposits that we mine today, The iron in the steel of bridges and buildings and the quiet interiors of machines were formed by ancient bacteria billions of years ago. These bacteria, called iron-oxidizing bacteria, absorbed dissolved iron from the early oceans and deposited it as solid iron oxide, layer by layer, over hundreds of millions of years, building up vast banded formations of iron-rich rock that now lie beneath our feet. When you cross a bridge, or hold a tool, or rest your hand on a railing, you may be touching something whose origin begins in a microbial life that ended long before the first animal ever swam or crawled or breathed. The bacteria that built those iron deposits are gone, but what they made endures, and we have shaped it into the architecture of our world. There are an estimated 10 nonillion bacteria on Earth. That is a 1 followed by 31 zeros. A number so vast it exceeds by far the total count of all the stars in the observable universe. The observable universe contains somewhere between 100 billion and 2 trillion galaxies, each holding hundreds of billions of stars. And still, the bacteria on this single, small, water-covered planet outnumber all of them. Let that settle into you like something warm and slow. The universe is enormous, vast beyond any honest comprehension. And yet this pale blue world, this fragile sphere drifting in one quiet corner of one ordinary galaxy, holds more individual living things than there are suns burning in all the skies of all the cosmos we can see. Life is not a small thing, even when the creatures that carry it are smaller than imagination. Bacteria live on every continent on this Earth, including Antarctica, where they survive beneath glaciers in near-freezing water, tucked into pockets of liquid that exist only because of the pressure of ice above them or the faint chemical warmth of ancient minerals below. Antarctica is the coldest, driest, most wind-swept place on the surface of this planet, a place that seems designed, in every possible way, to discourage life. And yet, bacteria are there. They are in the subglacial lakes that have been sealed from sunlight for millions of years. They are in the thin soils of the dry valleys where almost nothing else survives. They are in the rocks themselves, living just beneath the surface, warmed by the faint transmission of light through stone. Bacteria do not ask a place for permission before they arrive. They simply find a way to continue. Bacteria have been found thriving in the Mariana Trench, the deepest point on Earth, more than 36,000 feet below the surface of the Pacific Ocean, a place of absolute darkness, crushing pressure, and near freezing cold. At that depth, the pressure is more than 1,000 times greater than at sea level. It is a world that seems entirely inhospitable, and yet bacterial communities live there, feeding on the organic matter that drifts slowly down from the world above, that slow and constant rain of dead things that scientists call marine snow. The deepest place on Earth is not empty. It is not silent in the biological sense. It hums very quietly, with the slow and patient chemistry of microbial life, going about its ancient work in the cold and the dark and the deep pressure, unbothered by any of it. Bacterial life exists in clouds. Living cells have been collected from the atmosphere miles above the ground, drifting in the cold and turbulent air of the upper sky. Some scientists now believe that certain bacteria may not merely be passengers in clouds, carried there accidentally by wind, but active participants in weather itself, acting as nuclei around which ice crystals and water droplets form, seeding the conditions for rain. If this is true, then bacteria are not only witnesses to weather, they are, in some quiet and invisible way, its authors. The rain that falls on a summer afternoon, that taps at windows and soaks slowly into garden soil, may carry within it the legacy of bacteria that rode the wind for days, looking for somewhere to land. The sky is not as empty as it appears from below. Bacteria have been discovered living inside solid rock more than 2 miles beneath the surface of the Earth, in places so deep that no sunlight has ever reached them, where temperatures are high and oxygen is essentially absent, and where the only available food is the slow chemical energy locked inside the rock itself. These bacteria live on timescales almost impossible to reconcile with our own. Some are thought to divide only once every few thousand years. They are not in a hurry. They exist in a kind of geological patience, drawing sustenance from hydrogen and sulfur and the faint chemical gradients of stone, generation after generation, in absolute darkness, in absolute silence, in a stillness that makes our own quiet nights feel frenetic by comparison. There is life in the deep rock. It has always been there. Some bacteria live in highly acidic hot springs, like those found in Yellowstone National Park, where temperatures exceed 160 degrees Fahrenheit and the water is nearly as corrosive as battery acid. The springs at Yellowstone are alien landscapes, vivid with color, wreathed in steam, smelling of sulfur, surrounded by bare mineral earth that testifies to how thoroughly the water has dissolved everything that tried to grow too close. And in those springs, bacteria not only survive but thrive. They are the source of the vivid colors that ring the pools, the deep oranges, the bright yellows, the greens and blues that make Yellowstone's hot springs look like something painted by a dreaming hand. Those colors are life. They are microbial life, sitting serenely in water that would destroy almost any other living thing. Bacterial communities exist in the Dead Sea, one of the saltiest and most inhospitable bodies of water on Earth, a place so dense with dissolved minerals that most organisms cannot survive in it, that human bodies float effortlessly in it, that its name has become a metaphor for lifelessness itself. And yet, bacteria are there. Halophilic bacteria, salt lovers, have adapted to the extreme salinity, building cellular machinery that operates normally in conditions that would draw the water out of most cells and leave them shriveled and dead. They do not fight the salt. They have learned over immense stretches of evolutionary time to live inside it as naturally as we live inside air. Even the Dead Sea is not entirely without life. Even the most forbidding places carry within them some small and patient shimmer of biology. Bacterial communities exist in the upper atmosphere, where they drift in conditions of intense ultraviolet radiation, extreme cold, and near total desiccation, and where some scientists believe they may influence the patterns of rainfall and storm formation across the surface of the Earth below. The idea that living things participate in the making of weather is a profound one. We have always thought of weather as something that happens to life. Rain falls on us, wind moves around us, temperature shapes where we can live and how. But if bacteria seeding clouds can influence whether rain falls or doesn't fall, whether a storm forms or doesn't form, then life and weather are not separate things. They are braided together in a loop of influence that stretches from the deep soil to the high cold sky. Bacteria have been found living inside nuclear reactor cooling pools, environments saturated with ionizing radiation at levels that would be lethal to virtually any other living organism. The most famous of these radiation-resistant bacteria is Deinococcus radiodurans, sometimes called Conan the bacterium in scientific literature, a name given with genuine admiration. The radiation inside a reactor cooling pool would shatter DNA into fragments, and yet this bacterium reassembles its own broken genetic material with a precision and speed that scientists are still working to fully understand. It carries multiple copies of its genome, like a library with many redundant backups, and it repairs damage so quickly that the radiation can barely keep pace. It is, in the truest sense, nearly indestructible, and it lives in places where we build our most powerful machines. Every teaspoon of healthy garden soil contains between 100 million and 1 billion bacteria, a number so dense with life that the ground beneath a garden is, in a very real sense, more alive than it appears. When you press your hand into warm earth, when a child picks up a handful of soil and lets it fall between their fingers, they are touching one of the most densely populated ecosystems on the planet. The bacteria in that teaspoon are not all the same. They represent thousands of different species, different shapes and sizes and metabolic strategies, different relationships with the other organisms, the fungi, the nematodes, the tiny insects, that share that small and dark and rich underground world. Soil is not dirt. It is a living architecture built and maintained by creatures too small to see, going about their ancient work just beneath the surface of the ordinary world. Bacteria are prokaryotes, meaning they carry no nucleus. Their DNA floats freely inside them like a loose and quiet thread, unbound by any membrane, unenclosed in any separate chamber. In the cells of our own bodies and in all complex life, the genetic material is housed in a nucleus, a kind of inner room, sealed off and protected, where the instructions for life are kept carefully organized. Bacteria have no such room. Their DNA simply drifts within them, accessible and unguarded, like a song playing openly in an open space. This is one of the things that makes bacteria so ancient, so fundamental. They carry the most elemental possible version of a living cell, stripped of the elaborate internal architecture that came later. They are life at its most essential, and in that simplicity lies something close to perfection. A single bacterium is typically between 1 and 10 micrometers in length, so small that 1,000 of them could line up across a single human hair and still leave room. A micrometer is 1/1,000,000th of a meter. The period at the end of this sentence is roughly 500 micrometers wide. A bacterium sitting at one end of that period would be invisible to you, not just difficult to see, but genuinely completely invisible beyond the resolution of the naked eye. And yet within that unimaginably small space, a bacterium contains everything needed to be alive. The ability to take in energy, to build its own components, to respond to its environment, to repair itself, to reproduce. All of the machinery of life compressed into a space smaller than a whisper, smaller than a shadow, smaller than anything we can hold or touch or fully imagine. Bacteria have no mitochondria, the organelles that generate energy in the cells of more complex organisms, and instead produce their energy directly through their cell membrane, using it as a surface across which electrical gradients are built and released like a slow and steady tide. In our own cells, mitochondria are the power stations, separate, enclosed structures that burn fuel and release the energy that keeps us moving and thinking and breathing. Bacteria have folded this entire process into the membrane that forms their outer boundary. The wall of a bacterium is not merely a container. It is an active site of chemistry, a place where energy is born and distributed, where the cell's relationship with the world around it is continuously negotiated. In this way, every bacterial cell is its own power station, its boundary and its engine built into the same surface. Some bacteria have a whip-like tail called a flagellum that rotates like a tiny molecular propeller, allowing them to swim through liquid with a purposeful, directed motion that is beautiful to observe under a microscope. The bacterial flagellum is one of the most studied structures in all of microbiology, not only because of what it does, spin at extraordinary speed, driving the bacterium forward or backward through its watery world, but because of what it represents, a biological motor built from protein assembled by the cell itself, rotating at thousands of revolutions per minute. When a bacterium senses a gradient of food or light or chemical attraction, it spins its flagellum in one direction to move forward and reverses it to tumble randomly and reorient. It is not thinking, it is responding in the most elegant and ancient way to the pull of the world. Bacteria can form protective outer layers called biofilms, living communities wrapped in a shared gel-like shield of their own making, in which individual bacteria give up some of their independence in favor of the safety and stability of the collective. A biofilm is something between a colony and an organism. The bacteria within it communicate, divide labor, share resources, and protect each other from antibiotics and environmental stress in ways they could not manage alone. The plaque that a dentist gently removes from teeth is a biofilm. The slick surface of a stone in a stream is a biofilm. The thin living layer on the inside of a pipe, on the surface of a medical device, on the hull of a ship. These are all biofilms, quiet and persistent, built by bacteria that have decided in their wordless way that there is strength in staying together. Some bacteria produce endospores, dormant, armored versions of themselves, when conditions become too harsh to survive, encasing their essential genetic material in one of the toughest biological structures known to science. An endospore is not quite a bacterium and not quite a seed. It is a survival capsule, a distillation of the cell's most essential information wrapped in layers of protein and mineral so durable that endospores have been found to survive boiling radiation, years of desiccation, and the vacuum of outer space. When conditions improve, when water returns, when temperature normalizes, when nutrients become available, the endospore can germinate and a living bacterium emerges again as if from a long and untroubled sleep. This is one of the most astonishing biological tricks in the living world. The ability to become for a time almost nothing and then to become everything again. Bacteria communicate with each other through a chemical process called quorum sensing, releasing and detecting molecular signals that allow entire communities to coordinate their behavior, to act in certain ways as if they share a single distributed awareness, Each bacterium releases small molecules into its environment. As the population grows denser, the concentration of those molecules rises, and when it crosses a threshold, when the quorum is reached, the entire community shifts its behavior simultaneously. They might begin producing toxins or forming a biofilm or releasing light or launching an infection. They have been counting in a chemical sense without counting. They have been listening to each other in a language made of molecules. There is something in this that is worth sitting with quietly. The idea that even the simplest living things reach out to each other, that even bacteria do not entirely want to be alone. Some bacterial cells are capable of surviving complete dehydration, the total removal of water from their bodies, entering a state of suspended animation in which all biological activity ceases and from which they can return to full life when water becomes available again. Water is, for most life on Earth, an absolute requirement, the medium in which the chemistry of living things is dissolved and carried and transformed. Remove the water and life should stop, and for most organisms it does, permanently. But some bacteria replace their water with protective sugars that hold the shape of their cellular components in place, like a biological scaffold, preserving the architecture of the cell even in the total absence of moisture. They become, for a time, something closer to a mineral than an organism. And when the water returns, the scaffold dissolves back into life and the bacterium continues as if nothing happened. Bacteria lack a defined shape in some species. They can shift and morph depending on their environment, stretching or rounding in response to conditions around them, without the rigid cellular form that characterizes more complex organisms. Most bacteria have characteristic shapes, spheres called cocci, rods called bacilli, spirals called spirochetes, and these shapes are meaningful, influencing how the bacterium moves, how it interacts with surfaces, how it evades immune systems. But some bacteria are pleomorphic, capable of assuming multiple forms depending on temperature, nutrition, age, or chemical environment. They are not fixed, they are responsive. In this, they reveal something about the flexibility at the heart of life, the willingness of living things to change their shape in order to survive, to not be too attached to any single form, to remain at the deepest level, adaptable. The cell wall of many bacteria contains a unique molecule called peptidoglycan, a substance found nowhere else in all of nature, a molecular mesh that gives the bacterial wall its strength and its shape, and that serves as a target for some of the most important antibiotics in the history of medicine. Peptidoglycan is, in a sense, one of the signatures of bacterial life, a chemical marker that says this is a bacterium. It is a lattice of sugars and amino acids woven together into a net that surrounds the cell and holds it in form against the pressures of the world. When penicillin was discovered, the mechanism it exploited was the disruption of peptidoglycan, the unraveling of that net, the gentle dissolution of the bacterial wall. The wall opened. The bacterium, unable to hold its shape, came undone. It is a quiet chemistry, and it has saved hundreds of millions of lives. The human body contains approximately 38 trillion bacterial cells, a number roughly equal to the total number of human cells in the body, which means that in a very literal and measurable sense, you are as much bacterial as you are human. This is not a metaphor. This is biology. For every human cell in your body, there is approximately one bacterial cell accompanying it, living on your skin, in your gut, in your mouth, in the warm interior geography of your body, going about their own ancient work in the context of your life. You are, in the truest sense, a community. You are an ecosystem walking through the world. The boundaries of what we mean when we say the word yourself are softer and more populated than we tend to imagine when we think about who we are and where we end. The gut microbiome, the vast and varied community of bacteria living in the human digestive system, weighs between 2 and 5 pounds in a healthy adult, making it one of the largest organs in the body, if we are willing to extend the word organ to include something alive and distinct, something that is both part of us and not entirely us. That weight is not fat or fluid or fiber. It is life. Billions upon billions of individual organisms, thousands of species, living in the dark interior of the digestive tract, feeding on what we eat, producing compounds that our cells use, communicating with our nervous system, participating in the slow daily maintenance of our health. The gut microbiome is not a passenger. It is a partner, ancient, invisible, and in many ways essential to what it means to be a living human body. More than 1,000 different species of bacteria have been identified in the human gut, a diversity so rich it rivals some of the most complex ecosystems on the surface of the Earth. A coral reef, a tropical rainforest, a deep-sea vent community. These are the images we reach for when we want to conjure biodiversity, when we want to feel the overwhelming richness of life in a single place. And yet, inside the quiet interior of a human digestive tract, a comparable diversity has made its home. Each species occupies a different niche, feeds on different substrates, produces different compounds, interacts differently with the cells that line the gut wall and with the other species around it. The microbiome is not a monoculture. It is a complex and layered community, more intricate than we fully understand, and more important to our health than we are still in the process of discovering. Bacteria in the gut help digest foods that human cells alone cannot break down, including many of the plant fibers and complex carbohydrates that form the bulk of a healthy diet, converting them into compounds that the body can absorb and use. Our own digestive enzymes are powerful, but they have limits. There are entire categories of plant matter, the fiber in vegetables, the complex sugars in legumes and grains, that pass through the stomach and small intestine largely unchanged because our cells simply do not possess the tools to break them down. And then those fibers reach the large intestine where the bacteria are waiting. The bacteria produce the necessary enzymes, they ferment the fiber, they convert it into short-chain fatty acids that feed the cells lining the colon, that reduce inflammation, that nourish the body in ways that no human cell could accomplish alone. We digest, in part, by proxy. Gut bacteria produce vitamins, including B12, B2, and K. Essential nutrients that the human body absorbs and uses for everything from blood clotting to energy metabolism to the maintenance of healthy nerve function. We tend to think of vitamins as things we find in food, in the deliberate choices we make about what to eat, and they are. But they are also, in part, things we grow inside ourselves, in the warm and populated darkness of the intestines, where bacteria go about their chemistry and produce, as a byproduct of their own lives, compounds that our bodies have come to depend on. This is one of the quieter expressions of how thoroughly human health is entangled with microbial life. We do not simply tolerate the bacteria in our guts. We have built, over millions of years of coevolution, a relationship with them so close that we rely on their chemistry to sustain our own. The gut microbiome communicates with the brain through the vagus nerve, a long and branching nerve that connects the digestive system directly to the brain stem, influencing mood, stress responses, and behavioral tendencies in ways that scientists are only beginning to map. The vagus nerve is sometimes called the wandering nerve, and it wanders in the most literal sense, snaking from the brain down through the chest and into the abdomen, sending signals in both directions along its length. The bacteria in the gut produce neurotransmitters, serotonin, dopamine, GABA, the same chemical messengers that govern emotion and thought in the brain. They produce compounds that travel through the bloodstream to the brain. They influence the brain through the immune system. The relationship between gut and mind is not a metaphor, it is a physical and chemical highway, and the bacteria in your gut are, in some quiet way, participants in your inner life. Babies born vaginally are colonized by their mother's bacteria during birth, receiving in the very first moments of their lives outside the womb a seeding of microbial life that will shape the development of their immune system, their digestive function, and their long-term health in ways that researchers are still working to fully understand. The birth canal is not sterile. It is populated deliberately with lactobacillus and other bacteria that the mother's body has prepared as if in anticipation. As the baby moves through that passage, they are coated in a film of living organisms, a first gift, given without ceremony, without words, in the ancient and wordless language of biology. Those bacteria are among the first living things the baby encounters. They are the beginning of a microbiome that will spend a lifetime becoming its own complex and unique community. The skin hosts approximately 1,000 different bacterial species across its surface, a varied and dynamic community that forms one of the body's first and most important lines of defense against the pathogens that move through the world around us. The skin is not a passive barrier. It is a living ecosystem populated by bacteria that occupy different niches across the geography of the body. Different communities in the oily regions near the nose, in the moist warmth of the underarm, in the cooler and drier landscape of the forearm. These bacteria compete with harmful pathogens for space and resources, produce antimicrobial compounds, and signal the immune system about the state of the outer world. They are, in a very real sense, guards, not hired guards, not conscious guards, but ancient ones, whose presence on the surface of the skin has been part of the story of human health for as long as humans have existed. Bacteria in the mouth number in the billions, with more than 700 species identified across gums, teeth, tongue, and the moist interior surfaces of the cheeks, making the oral cavity one of the most densely and diversely populated microbial environments in the human body. The mouth is a warm, nutrient-rich, constantly refreshed environment, exactly the kind of place that microbial life finds irresistible. Most of the bacteria in the mouth are harmless or beneficial, producing enzymes, competing against pathogens, maintaining the delicate balance of the oral environment. Some, however, under the right conditions, excess sugar, disrupted saliva, poor hygiene, can form biofilms on tooth surfaces that produce acid and contribute to decay. The mouth is, in this sense, a negotiation, a balance between bacterial communities that is always in motion, always responding to what we eat and drink and do. Disrupting the microbiome through overuse of antibiotics has been linked in research studies to long-term changes in immune function, susceptibility to certain infections, and alterations in mood and mental health that can persist for months or years after the antibiotic course has ended. Antibiotics are among the greatest medical achievements in human history. They have saved hundreds of millions of lives, and the world before them was darker and more fragile than most of us now living can easily imagine. But they are imprecise instruments. They do not distinguish between the harmful bacteria they are targeting and the beneficial ones they destroy along the way. A course of antibiotics is, for the microbiome, a kind of weather event, a disruption that passes but leaves the landscape changed. The community rebuilds, but it may not rebuild exactly as it was, and in those differences, subtle and slow, some of our health follows. Some bacteria can double their entire population in as little as 20 minutes under ideal conditions, a rate of growth so rapid that a single bacterium, given unlimited nutrients and space and time, could theoretically produce a mass of descendants larger than the observable universe within a few days. This is not something that actually happens, of course. Resources run out, space fills, competition intensifies. But the potential is real, and it speaks to something extraordinary about the nature of bacterial reproduction, the power of exponential growth, the way that small things, doubling and doubling again, can become incomprehensibly large in a very short time. A single bacterium at dawn, dividing every 20 minutes, becomes over 68 billion by nightfall. The mathematics of doubling is one of the quietest and most powerful forces in all of biology. Deinococcus radiodurans is considered the world's most radiation-resistant organism, capable of reassembling its own shattered DNA after exposure to levels of radiation thousands of times greater than the dose that would kill a human being. This bacterium carries, within its tiny body, one of the most extraordinary repair systems in all of known life. Its genome can be shattered into hundreds of fragments by intense radiation, literally broken into pieces. And within a matter of hours, the bacterium locates every fragment, recognizes its correct position in the sequence, and stitches the genome back together with an accuracy that approaches perfection. Scientists are studying this repair mechanism not only out of wonder, but out of hope. Understanding how Deinococcus radiodurans fixes its DNA may one day help us find new ways to treat the DNA damage that underlies cancer and aging. Certain bacteria produce bioluminescence, a cold, soft, living light generated through chemical reactions within their cells. And in places where they gather in vast numbers, they can make the surface of the ocean glow with a blue or green shimmer that has been observed and wondered at by sailors and scientists for centuries. Bioluminescent bacteria live in the gut of certain fish, in the flesh of decaying wood and meat, in the open water of warm tropical seas. When a wave breaks on a bioluminescent shore, the water glows at the point of impact, as if the sea itself is briefly illuminated from within. These bacteria produce light not for beauty, not for any purpose that maps onto human intention, but as a byproduct of their own chemistry, and in doing so, they make the dark parts of the world briefly, quietly, astonishingly bright. Some deep-sea bacteria survive without sunlight, deriving energy not from photosynthesis, but from the chemical energy locked in hydrogen sulfide and other compounds that vent from cracks in the ocean floor, a process called chemosynthesis, which suggests that life does not require a star in order to exist. This is one of the most profound implications of deep-sea microbiology. Sunlight is the engine of almost all life on Earth, the energy that plants capture and that passes through every food chain, from grass to deer to wolf, from algae to krill to whale. But chemosynthetic bacteria bypass all of that. They live in complete darkness, drawing their energy from the chemistry of the Earth itself. Their existence has changed our understanding of where life might be found in the universe, on ocean worlds without suns, on moons with liquid interiors, in dark and pressurized places we have not yet visited. Bacteria can transfer genetic material to one another without reproducing, a process called horizontal gene transfer, allowing them to share survival traits, including antibiotic resistance, almost instantaneously across species and even across very different types of bacteria. In most organisms, genetic information travels vertically, from parent to offspring, down through the generations. Bacteria can also pass genes sideways, directly from one cell to another, using tiny packages of DNA called plasmids, or through viruses that carry genetic material between them. This means that a bacterium that develops resistance to an antibiotic can, in a sense, teach that resistance to its neighbors, even to bacteria of entirely different species, within a single generation. It is a form of genetic generosity that has enormous implications for medicine, and it is one of the reasons why antibiotic resistance spreads through bacterial populations with a speed that continually challenges our ability to keep up. Some bacteria produce natural antibiotics to defend themselves against competing microbes, secreting chemical compounds into their environment that kill or inhibit the growth of bacterial neighbors in a kind of chemical warfare that has been going on in the soil and in living bodies for hundreds of millions of years before any human being thought to harness it. The penicillin that Alexander Fleming discovered in 1928 was not created by him. It was being produced by a mold, Penicillium, as part of its own ancient strategy for surviving in a competitive microbial world. When scientists began looking for new antibiotics, they turned to the soil, to the bacteria living there, and found that the soil was already full of them, already populated with organisms that had been producing antibiotic compounds for far longer than we have been looking. The medicines that have saved the most human lives were, in a very real sense, already there, waiting to be recognized. Magnetotactic bacteria contain tiny magnetic crystals inside their cells, structures called magnetosomes made of magnetite or greigite, and they use these internal compasses to orient themselves along the lines of the Earth's magnetic field, navigating through sediment and water in ways that most bacteria cannot. This is one of the most quietly extraordinary features in all of microbiology, a bacterium with a compass. The magnetosomes are arranged in chains within the cell like beads on a thread, and they align with the Earth's magnetic field so precisely that magnetotactic bacteria in the northern hemisphere swim north and downward, toward the sediment-rich bottom waters where they find the chemical conditions they prefer. They do not think about where they are going. They are pulled gently and continuously by the field of the planet itself, drifting along invisible lines of force that thread through the world in every direction. Certain bacteria can break down oil spills, consuming petroleum compounds as a food source, metabolizing the complex hydrocarbons in crude oil into simpler, less harmful compounds, in a process that scientists have been studying and attempting to harness as a tool for environmental cleanup. After the Deepwater Horizon oil spill in 2010, researchers discovered that bacteria native to the Gulf of Mexico responded to the influx of oil not with harm, but with an explosion of population growth, consuming the oil as food, forming blooms of microbial life in the oiled water, working at a pace and scale that surprised even the scientists studying them. The ocean had, in a sense, its own cleanup crew, ancient, invisible, already present, waiting for exactly this kind of chemical abundance. Bacteria have been metabolizing hydrocarbons for longer than petroleum has existed as an industry. They were ready for us before we arrived. Certain bacteria can extract metals from rock through a process called bioleaching, a commercial mining application in which bacteria are used to dissolve and concentrate copper, gold, uranium, and other metals from ores that would be too low grade or too costly to process by purely chemical or mechanical means. Thiobacillus ferrooxidans and similar bacteria oxidize the sulfide minerals surrounding metal deposits, releasing the metals into solution and allowing them to be collected and refined. This is biology doing what chemistry does, but more slowly, more selectively, and in some cases more efficiently. The copper in your phone, the gold in your electronics. Some of it began its journey from ore to object in the company of bacteria, which dissolved the rock around it and offered it up to the processes of human industry. The oldest organisms on Earth are in this way participants in the newest economies. Some bacteria can survive in the vacuum of space for short periods, sealed inside the protective shell of meteorite rock, a fact that has given genuine scientific weight to the theory of panspermia, the idea that life, or the seeds of life, may be capable of traveling between worlds on the backs of rocks flung from one planet to another by the violence of collision and eruption. Experiments have exposed bacterial spores to the vacuum and radiation of space attached to the exterior of spacecraft and found that some survive, not indefinitely, but long enough to raise the question of whether a journey from Mars to Earth or from Earth to Mars is something biology could endure. The distances are vast, the conditions are brutal, but bacteria have surprised us before with what they are capable of enduring. They may yet surprise us about how far life can travel and where it might land. Bacteria are responsible for the nitrogen cycle, the great planetary process by which nitrogen from the atmosphere is converted into a form that plants can absorb and use to build proteins, and then returned to the air again when organisms die and decompose. Without bacteria to perform this cycle, the nitrogen in the atmosphere would remain locked away in a form that plants cannot access, and without usable nitrogen, plants cannot make the proteins that animals need to eat. The nitrogen cycle is not a metaphor for interdependence. It is a literal, chemical, ongoing process without which agriculture would collapse, without which forests would thin and die, without which the food that sustains every animal on Earth would cease to exist. Bacteria are not merely participants in the nitrogen cycle, they are its essential engine, and they have been running it without interruption for billions of years. Rhizobium bacteria live inside the root nodules of legume plants, in beans, clover, peas, alfalfa, and convert atmospheric nitrogen into ammonia, a form that plants can absorb and use to grow. This relationship is one of the oldest and most beautiful partnerships in the living world. The plant provides the bacteria with sugars produced through photosynthesis, energy captured from sunlight. The bacteria provide the plant with fixed nitrogen, fertility captured from the air. Each gives what the other cannot make for itself. Farmers have known for centuries, even before they understood the microbiology, that planting legumes in a field replenishes it, that there is something in these plants that gives back to the soil what other crops take away. What they were observing was the invisible labor of rhizobium, going about its chemistry in the quiet interior of root nodules, turning air into fertilizer, sustaining agriculture one small cellular reaction at a time. Bacteria decompose dead organisms, returning the carbon, nitrogen, phosphorus, and other essential elements locked in their bodies back to the soil and the water, where they can be taken up again by the living, closing the great elemental cycles that allow life on Earth to continue without consuming itself. Every living thing that dies is a gift to the bacteria of the soil. Every fallen leaf, every drowned insect, every body of every creature that has ever lived becomes, in the chemistry of bacterial decomposition, raw material for the next generation of life. This is not a grim fact. It is one of the most elegant and generous features of the living world, the way that death is not an ending but a transformation, a returning, a redistribution of the materials of life back into the great slow river of biology. Bacteria are the agents of that return. They are the Earth's own composters, its quiet alchemists, its faithful recyclers. The smell of rain on dry earth, that particular beloved scent that has been given the name petrichor, is partly produced by a soil bacterium called Streptomyces, which releases a compound called geosmin into the air when rain disturbs the soil, and which the human nose detects with extraordinary sensitivity, at concentrations as low as a few parts per trillion. We are more sensitive to geosmin than we are to most other odors. Some researchers speculate that this sensitivity is ancient, that it evolved as a way for our ancestors to detect the presence of moisture in dry landscapes, to know that water was near. If that is true, then the pleasure we feel when rain begins to fall on summer Earth is not merely aesthetic, it is biological. It is the echo of a very old need, answered by the chemistry of a bacterium that lives in the soil and has been living there for a very long time. Ocean bacteria produce approximately 50% of the world's oxygen, an amount equal to all the oxygen produced by all the forests and grasslands and plants on land combined, making them, by any measure, the most important photosynthesizers on this planet. Prochlorococcus, a tiny cyanobacterium discovered only in 1988, is believed to be the most abundant photosynthetic organism on Earth, with a population in the trillions of trillions, spread across every sunlit ocean on the planet. Every other breath you take, the oxygen in your lungs as you exhale gently into sleep tonight, was produced in the ocean by bacteria too small to see, going about their work in the warm and light-dappled surface water of a sea you may never visit. The oceans breathe for us, and the bacteria breathe for the oceans. Bacteria in wetlands produce methane as a byproduct of the decomposition of organic matter in low oxygen conditions, a process that contributes to the natural greenhouse effect and has been part of the Earth's climate regulation for millions of years, long before industrialization added its own layer to the story. Methane-producing bacteria, called methanogens, thrive in the anaerobic depths of swamps and marshes, in the waterlogged soils of rice paddies, in the digestive systems of ruminant animals, in the deep sediments of the ocean floor. They are among the most ancient bacteria known, representing a lineage that stretches back to a time when the Earth's atmosphere was very different from what it is today. They do not produce methane as a deliberate act. It is simply the exhaust of their metabolism, a compound that is released as they feed and grow, as all living things release something into the world around them in the course of being alive. Some bacteria form symbiotic relationships with deep-sea tube worms, living inside the bodies of these creatures and providing them with all of their nutrition through chemosynthesis, in an ecosystem so remote and dark and pressurized that sunlight has never touched it, and where all of the energy that sustains life comes from the chemistry of the Earth itself. The tube worms around hydrothermal vents can grow to enormous sizes, meters long, waving in the superheated water like pale red flowers. And they have no digestive system of their own. They do not eat. They do not filter food from the water. They simply house bacteria in specialized organs, and the bacteria do all the metabolic work, converting hydrogen sulfide into energy and sharing it with their host. The tube worm and the bacterium are each the other's world, a closed and quiet partnership burning in the dark at the bottom of the sea. Bacteria are the primary decomposers in the deep ocean, breaking down the remains of organisms that drift down from the sunlit surface in what oceanographers call marine snow, a perpetual, slow, delicate rain of organic particles that falls through the water column and sustains the deep-sea food web. Marine snow is one of the most peaceful images in all of oceanography. The endless, soft descent of organic material through the dark water, like snow in slow motion, carrying with it the carbon and nitrogen and energy of organisms that lived and died far above. Bacteria attach to these particles as they fall or wait for them on the seafloor and break them down into their chemical components, releasing nutrients that cycle back up through the water column, completing a loop of carbon and life that connects the surface of the ocean to its darkest depths, mediated at every step by the chemistry of bacteria. Cyanobacteria in the ocean form massive blooms during warm seasons, expanses of microbial life so large that they are visible from space, stretching across hundreds of miles of ocean surface in colors of green and turquoise and rust, a testament to the scale at which bacterial life can operate when conditions align in its favor. These blooms are not merely impressive to observe from orbit, they are ecologically significant, producing vast quantities of oxygen, fixing nitrogen, feeding vast food chains. They are also, in some cases, a warning, Certain cyanobacterial blooms produce toxins harmful to fish and mammals, including humans, and the warming and nutrient enrichment of ocean waters can make blooms more frequent and more intense. Even at the scale of satellite imagery, the story of bacteria is a story of balance, of the thin and ancient equilibrium between the abundance of microbial life and the health of the larger world it sustains. Without soil bacteria, topsoil would stop forming, and without topsoil, the agriculture that feeds 8 billion human beings would, over time, become impossible. This is not hyperbole, it is ecology. Topsoil takes hundreds of years to form through the patient chemistry of bacterial decomposition, the binding of mineral particles by bacterial and fungal secretions, the aeration and structuring of the soil by the organisms that live within it. Remove the bacteria, and the process stops. The soil becomes inert, mineral but not alive, capable of supporting structure but not capable of sustaining growth. Every crop that has ever been grown in any field anywhere on Earth was grown in soil that bacteria made and maintained and continue to make and maintain at this very moment in the dark beneath our feet, invisibly, without recognition, as they have always done. Only a tiny fraction of bacterial species, fewer than 1% of all known bacteria, are capable of causing disease in humans. This is a fact worth sitting with in the quiet of the night, when the word bacteria might carry darker connotations. The vast, overwhelming majority of bacteria that exist on and in and around us are neutral or beneficial, cycling nutrients, maintaining the microbiome, producing vitamins, protecting against pathogens, decomposing waste, sustaining ecosystems. The handful of species that have learned to exploit human biology for their own reproduction represent an extreme minority within a kingdom of extraordinary diversity. The story of bacteria is not a story of threat. It is a story of coexistence, of partnership, of a world in which we are continuously surrounded by microbial life that asks nothing of us and offers, in its quiet way, nearly everything. The bubonic plague, which swept through Europe and Asia in the 14th century and killed an estimated 1/3 of Europe's entire population, was caused by the bacterium Yersinia pestis, a tiny, rod-shaped organism transmitted through the bites of fleas that lived on rats and which found in the crowded and connected cities of the medieval world conditions perfectly suited to its own rapid spread. The plague reshaped history in ways that echoed for centuries. It emptied cities, collapsed feudal economies, contributed to social upheaval, and altered the genetic makeup of European populations in ways that researchers are still tracing. All of this from a bacterium, something invisible to the naked eye, something that has no intention and no awareness, that simply was and replicated and moved through a world that had no way to stop it. The scale of human history has more than once pivoted on the behavior of something too small to see. Tuberculosis, caused by the bacterium Mycobacterium tuberculosis, remains one of the world's leading infectious disease killers today, infecting an estimated 10 million people each year and claiming more than 1 million lives annually, making it, despite centuries of medical advancement, still one of the most persistent and devastating bacterial diseases in human history. Mycobacterium tuberculosis is, in a terrible way, a remarkable organism. It has evolved the ability to survive inside the very immune cells that are sent to destroy it, sheltering inside macrophages and lying dormant for years or decades before becoming active again. It is carried by roughly 1/4 of the world's population in its latent form, present but not active, waiting. It is ancient. Evidence of tuberculosis has been found in Egyptian mummies and in human bones from thousands of years ago. It has been with us for a very long time, and it remains with us still. Helicobacter pylori, a spiral-shaped bacterium that lives in the acidic environment of the human stomach, was discovered in 1983 to be the primary cause of most peptic ulcers, a discovery so counterintuitive, so contrary to the prevailing medical understanding, that the researchers who made it, Barry Marshall and Robin Warren, had to perform extraordinary experiments to be believed, and ultimately received the Nobel Prize in Physiology or Medicine in 2005 for their work. Before Marshall and Warren, peptic ulcers were believed to be caused by stress, by spicy food, by lifestyle, conditions that no antibiotic could treat. After their discovery, the treatment for most ulcers became a course of antibiotics. The shift was profound. A condition that had caused chronic suffering for millions of people, that had generated entire industries of antacids and dietary advice, was revealed to be an infection, treatable and curable. once someone was willing to ask a different question. Certain strains of Escherichia coli, more commonly known as E. coli, are completely harmless and essential to human gut health, living in the large intestine and contributing to the normal function of the digestive microbiome, despite the bacterium's reputation in headlines as a source of foodborne illness. E. coli is one of the most studied organisms in all of biology, a workhorse of the laboratory, a model organism whose genetics and biochemistry have been mapped in extraordinary detail, a bacterium whose cooperation with science has produced countless medical and biological discoveries. The strains that cause illness represent a small fraction of E. coli diversity. The strains that live peacefully in the gut, that help metabolize nutrients and maintain the microbial balance of the intestines, are quiet contributors to the daily function of health that most people carry without ever knowing it. Lactobacillus bacteria, found in yogurt, kefir, sauerkraut, kimchi, and many other fermented foods, support the health of the gut lining and the immune function of the intestinal system, and have been consumed by human beings in one fermented form or another for thousands of years, long before anyone understood the microbiology of why these foods seemed to nourish and strengthen. Fermentation is one of humanity's oldest food technologies, and it was always, in its essence, a collaboration with bacteria. The tang of yogurt, the sourness of kimchi, the complex depth of aged cheese, these flavors are the signatures of bacterial activity, the sensory record of a metabolic transformation that bacteria have been performing on organic matter since long before humans began harvesting it. We did not invent fermented food. We discovered that bacteria were already fermenting things, and we learned to guide that process. Certain bacteria produce toxins of almost incomprehensible potency, including botulinum toxin produced by Clostridium botulinum, which is the most toxic biological substance known to science, with a lethal dose in humans measured in nanograms per kilogram of body weight. A nanogram is 1/1000th of a gram, the power contained within something invisible to the naked eye, within something that has no intention and no malice, that simply produces this compound as a byproduct of its own metabolism, is one of the more quietly astonishing facts in all of biology. And yet botulinum toxin, in extraordinarily small and carefully controlled medical doses, is used therapeutically to treat muscle spasticity, chronic migraines, excessive sweating, and, in its most widely known cosmetic application, to smooth the expression lines of the human face. The most toxic substance in the world has become, in human hands, a medicine. The antibiotic penicillin, the discovery that inaugurated the modern age of antibiotics and that has saved an estimated 200 million human lives since its introduction, was found accidentally by Alexander Fleming in 1928 when he noticed that a mold contaminating one of his bacterial cultures had killed the bacteria around it. The mold was producing a compound that the bacteria could not survive. Fleming named the compound penicillin. What he was observing was not a laboratory accident. It was, in a deeper sense, the surface of an ancient competition, the chemical warfare between molds and bacteria that had been going on in the soil and on decaying organic matter for hundreds of millions of years. Penicillin did not come from a laboratory. It came from a living thing, defending itself as living things do. Fleming simply had the presence of mind to notice that it was happening and to ask why. Antibiotic resistance is driven by the extraordinary speed and flexibility of bacterial evolution. Strains that survive exposure to antibiotics pass on their resistance genes to their descendants and, through horizontal gene transfer to neighboring bacteria as well, creating an accelerating challenge that medical science is continuously working to understand and address. Every time an antibiotic is used, it creates selective pressure. The bacteria that cannot survive it die, and the bacteria that can survive it reproduce. Over generations measured in minutes rather than years, the resistant bacteria come to dominate the population. This is not a failure of medicine. It is an expression of one of the most fundamental forces in biology, the relentless, patient, entirely undirected drive of life to continue, to find a way, to adapt to whatever the environment requires. Bacteria do not intend to resist our medicines. They simply are very good at being alive. Fecal microbiota transplants, the transfer of carefully screened bacterial communities from the gut of a healthy donor into the gut of a patient whose own microbiome has been disrupted, have shown remarkable success in treating recurrent Clostridioides difficile infections, a condition that can be life-threatening and that often resists conventional antibiotic treatment. The idea sounds, at first encounter, startling, but the logic is elegant. If a patient's microbiome has been devastated and cannot recover on its own, the most efficient way to restore it may be to transplant a healthy, established microbial community from someone else, to reseed the ecology of the gut the way a forest might be reseeded after a fire. The results have been striking enough that fecal microbiota transplantation is now a recognized medical treatment, and researchers are actively exploring its potential for a wide range of conditions connected to microbiome health. The microbiome can be given, it can be shared. It is, in a sense, a kind of medicine grown inside us. Bacteria were the first organisms used to produce human insulin for people with diabetes, through genetic engineering first achieved in the 1970s, a development that transformed the treatment of diabetes and opened the door to an entire field of biotechnology in which living organisms are engineered to produce medicines that human bodies need. Before recombinant insulin produced in bacteria, diabetics relied on insulin extracted from the pancreases of pigs and cows, a supply that was expensive, difficult to scale, and occasionally caused immune reactions in human patients. When scientists inserted the human insulin gene into Escherichia coli, the bacteria began producing human insulin as naturally as they produce any other protein. The bacteria did not know they were producing medicine. They were simply reading the instructions encoded in their new DNA and doing what bacteria always do, expressing genes, building proteins, going about their ancient and patient work. The polymerase chain reaction, the technology behind COVID-19 tests, DNA forensics, paternity testing, archaeological genetics, and a vast range of modern biological research relies on an enzyme originally isolated from a heat-loving bacterium called Thermus aquaticus, discovered living in the hot springs of Yellowstone National Park. The enzyme, called Taq polymerase, is stable at the high temperatures required to separate DNA strands for copying, temperatures that would destroy the equivalent enzymes from most other organisms. Without a heat-stable DNA polymerase, PCR would not work. Without Thermus aquaticus, there would be no Taq polymerase. The bacterium living in a hot spring in Wyoming, going about its ancient chemistry in water too hot for almost any other life, was quietly carrying the enzyme that would one day become the foundation of modern molecular biology. It was there before we needed it. It was always there. Scientists are developing bacteria-based systems for the production of biofuels, using engineered bacterial metabolism to convert plant sugars, agricultural waste, and even carbon dioxide into ethanol and other liquid fuels that could one day reduce our dependence on petroleum. The idea is to harness the chemical capabilities that bacteria have already evolved, their ability to ferment sugars to produce organic compounds, to work efficiently at the molecular level, and redirect those capabilities towards the production of fuels. Some bacteria can produce hydrogen gas as a metabolic byproduct. Others can ferment cellulose from plant waste into ethanol. Others still are being engineered to capture carbon dioxide from the atmosphere and convert it into useful chemicals. Bacteria have been running their own chemistry for billions of years. We are only just beginning to learn how to ask them, very carefully, to run ours. Bacteria are used in wastewater treatment plants around the world to break down human waste and the chemical contaminants in sewage, converting them into simpler, less harmful compounds and helping to purify the water before it is returned to rivers, lakes, and eventually the water cycle that sustains all life. In this, bacteria are doing for our waste what they have always done for the waste of every organism that has ever lived on Earth: decomposing it, metabolizing it, cycling the elements it contains back into the world in forms that the living world can use. The sewage treatment plant is, in a sense, a managed version of something bacteria have been doing naturally for billions of years. A controlled ecology, curated by engineers and managed by microbiologists, but ultimately driven by the same ancient chemistry that breaks down the fallen leaf and the dead creature in the soil. We have learned to invite bacteria into our infrastructure. We could not do without them there. Certain bacteria are being studied for their remarkable ability to absorb and concentrate heavy metals, lead, mercury, arsenic, cadmium, from contaminated soil and water, offering a potential approach to environmental remediation that uses living chemistry to undo some of the damage that industrial activity has done to the landscape. This process, called bioremediation, is already being used in some contaminated sites where specific bacterial communities are introduced or encouraged to grow in the presence of pollutants and gradually remove them from the environment. It is a slow process, measured in months and years rather than days, but it is often more thorough, more selective, and less disruptive to the surrounding environment than purely chemical or mechanical cleanup methods. Bacteria are being asked to clean up after us, and they are, in their patient and chemical way, doing so. Bacteria have been engineered to produce spider silk proteins, the material in spider dragline silk, which is one of the strongest and most flexible natural materials known to science, with a tensile strength exceeding that of steel at a fraction of the weight. Spiders cannot be farmed for silk the way silkworms can. They are territorial and cannibalistic in close quarters. But their silk genes can be extracted and inserted into bacteria, which then produce the silk proteins in large quantities during fermentation. The potential applications range from bulletproof materials to surgical sutures to artificial tendons and ligaments. Bacteria are being asked to produce, at scale, a material that evolution took millions of years to design in the body of a spider, and they produce it as readily as they produce any other protein, in ordinary growth medium, in tanks, without fanfare. CRISPR-Cas9, the revolutionary gene editing technology that has transformed biology and medicine and that has been used to edit the genomes of crops, animals, and human cells, was discovered not in a laboratory but in the immune systems of bacteria, where it functions as a natural defense against viral infection, remembering the genetic sequences of viruses and using molecular scissors to cut and destroy viral DNA if it appears again. The bacteria had invented genetic memory. They had invented a system for recognizing molecular threats from the past and responding to them with chemical precision. When scientists understood what CRISPR was doing inside bacteria, they realized that the same molecular mechanism could be repurposed, that the scissors bacteria used to cut viral DNA could be directed with extraordinary precision to cut any DNA sequence in any organism. The greatest biotechnology tool of our era was already there in bacteria, running quietly, waiting to be understood. Bacteria are responsible for the transformation of raw ingredients into some of the most beloved and complex foods in human culture. Yogurt and cheese and sourdough bread and kimchi and sauerkraut and miso and vinegar and soy sauce and many others. Foods whose flavor, texture, and preservation depend entirely on the metabolic activity of bacterial communities working in the quiet dark of a jar or a barrel or a fermentation vessel. Fermentation is one of the oldest relationships between human beings and bacteria, older than agriculture, older than writing, older than the first cities. Long before anyone understood that bacteria existed, humans observed that milk left in the right conditions would sour and thicken, that cabbage packed with salt would transform over weeks into something sharp and preserved, that dough left overnight would rise in the morning with a new and complex fragrance. They could not see what was doing this, but they could taste the result, and they kept doing it. Scientists are exploring the possibility of engineering bacteria to seek out and destroy cancer cells from within the human body, bacteria that can penetrate the oxygen-poor interior of solid tumors, where conventional drugs often cannot reach, and deliver therapeutic compounds directly to the cancer cells they find there. Some bacteria naturally colonize tumors, drawn by the chemical environment of rapidly dividing cells and the low oxygen conditions that characterize tumor interiors. Researchers are studying how to load these bacteria with cancer-killing compounds, how to engineer them to recognize cancer cells specifically, and how to control their activity safely once they are inside the body. The idea of a living medicine, a bacterium engineered to navigate the interior of the body and find what needs to be found, is one of the more extraordinary frontiers in modern medicine. It is biology being asked to heal biology at the smallest possible scale. Bacteria have been engineered to produce biodegradable plastics, specifically a family of polymers called polyhydroxyalkanoates, which they accumulate inside themselves as energy storage compounds under conditions of nutrient stress, and which can be harvested and processed into a plastic that breaks down naturally in soil and water rather than persisting for centuries like conventional petroleum-based plastic. The bacteria produce these plastics using the same fermentation processes that produce beer and yogurt, converting sugars or plant waste into polymer chains, storing them as granules within the cell, like tiny biological factories. The plastic that comes out of this process is flexible, strong, and genuinely biodegradable. It is one of the more elegant possible answers to a problem that conventional chemistry has not yet solved: how to make the plastics we rely on without leaving behind a material that will outlast everything we use it for. Thiomargarita namibiensis, discovered off the coast of Namibia in deep-sea sediments, is the largest bacterium ever found, visible to the naked eye at up to 3/4 of a millimeter wide, a sphere of living chemistry large enough to be seen without any instrument, sitting at the outer edge of what we thought possible for a single bacterial cell. For most of the history of microbiology, bacteria were defined by their invisibility, by the fact that they existed below the threshold of human perception. Thiomargarita namibiensis broke that definition. It sits in the sediment like a tiny pearl, filled with a large internal vacuole of nitrate-rich water, its living material arranged in a thin layer around the periphery of the cell. It does not move. It does not have a flagellum. It simply sits, enormous and still, a bacterium at the edge of visibility, like a quiet argument against every assumption about how small living things are supposed to be. Some bacteria form long multicellular chains that glide together in coordinated movements across surfaces, not swimming, not tumbling, but sliding in smooth and purposeful arcs, like a slow river of living cells finding its way across the microscopic landscape of soil or plant matter or the surface of a leaf. Myxobacteria are among the most socially complex bacteria known. They communicate with each other chemically, coordinate their movements, hunt prey together by surrounding it with a wave of advancing cells, and when food becomes scarce, they aggregate into complex multicellular structures called fruiting bodies. Tiny, differentiated structures that look, under a microscope, remarkably like the fruiting bodies of fungi, with stalk cells that sacrifice themselves so that spore cells can be carried aloft to new environments. They are bacteria. They are also, in their quiet way, something more than we expect bacteria to be. Certain bacteria produce pigments of vivid and extraordinary color: deep purple, electric blue, bright orange, burning red, which they use as chemical defenses, as sunscreens against ultraviolet radiation, as signals to other organisms, or simply as metabolic byproducts of chemistry that turns out to have beautiful side effects. The pink and orange colors of flamingos come from the bacteria and algae they eat. The deep purple of some sea snails comes from a compound related to bacterial pigments. The blue-green shimmer of some exotic cheeses is the work of penicillium bacteria and their relatives. Bacteria color the world around us in ways that we rarely credit them for. The vivid rings of color in Yellowstone's hot springs, the seasonal red tides in coastal oceans, the bright orange biofilms on acidic rock. Color in the natural world is often a sign that life is present, and very often that life is bacterial. The bacterium Ideonella sakaiensis, discovered in 2016 in soil near a plastic bottle recycling facility in Japan, can break down PET plastic, the material used in most plastic bottles, and digest it as a food source, using it as its primary carbon and energy supply in the absence of other nutrients. This discovery astonished scientists not only because of what the bacterium can do, but because of how recently it apparently evolved this capability. PET plastic has only existed since the 1940s. That a bacterium could evolve the enzymes needed to degrade an entirely synthetic material within a few decades of that material appearing in the environment is a demonstration of the speed and flexibility of bacterial evolution that researchers found both remarkable and, perhaps, hopeful. Where we have left plastic, bacteria are learning to eat it. Where we have left a chemical problem, biology is, very slowly and very patiently, beginning to find an answer. Some bacteria have been found living inside gemstones, within tiny pockets of ancient fluid trapped in quartz and other crystals during their formation, sealed inside for millions of years, possibly sustained by the slow chemistry of mineral dissolution in the trapped water. The idea of life inside a gemstone is one that sits at the border between geology and biology, a reminder that the boundaries between the living and the non-living world are often softer and more porous than we expect. The crystals carry within them, sealed in their mineral clarity, a record of the ancient environment in which they formed. the temperature of the water, the chemistry of the minerals, and apparently sometimes the life that was present when the crystal closed around its small interior world. A gem is not only beautiful, it is, in the right light, a window into a very old and very quiet kind of life. Bacteria in the gut of certain termites allow them to digest wood, breaking down the cellulose in plant fiber into sugars that the termite can absorb and use for energy, in a partnership so essential that without their microbial inhabitants, termites could eat wood all day and derive nothing from it, and would eventually starve. Termites are, in a very real sense, walking fermentation vessels, creatures whose ability to exploit one of the most abundant food sources on Earth depends entirely on the bacteria and other microorganisms they carry within them. The termite provides shelter and a stream of food. The bacteria provide the enzymes that transform that food into something nourishing. Neither organism could perform the function without the other. This is one of the most studied and best understood examples of how, throughout the history of life on Earth, complex organisms have built their capabilities not by evolving every function themselves, but by forming alliances with microbes that already had what they needed. Some marine bacteria are so exquisitely sensitive to light that they adjust their photosynthetic behavior based not merely on the presence or absence of sunlight, but on the specific color and wavelength of the light they detect, tuning their pigments and metabolic activity to the precise quality of the light that filters down through the water column at different times of day and at different ocean depths. This is a degree of environmental sensitivity that we might more naturally associate with complex animals. The kind of nuanced perception that we imagine requires a nervous system, a brain, an experience of the world. And yet, bacteria perform it with molecular machinery, with photoreceptors and pigments and chemical signaling without any of the apparatus of consciousness. They sense the color of light. They respond to the quality of the day. They are, in this sense, paying a kind of attention to the world around them, not with awareness, but with the deep and patient chemistry of life. Certain bacteria produce electrically conductive protein filaments called microbial nanowires, hair-like appendages that can transmit electrons across distances many times the length of the bacterial cell itself, allowing bacteria to effectively breathe by sending electrons out through these wires to distant electron acceptors in the environment. This discovery has fascinated engineers and materials scientists as much as microbiologists. A bacterium that conducts electricity, that extends a living wire into the world and uses it to carry electrical charge from its metabolism to the minerals around it, is a biological solution to an engineering problem that we solve with copper and silicon and insulation. Researchers are studying microbial nanowires as potential components in biological electronics, in sensors, in systems that connect the living world to the electrical world in ways we have not yet imagined. The bacterium has been doing this in the mud for millions of years. We are only now learning to appreciate it. Some species of bacteria can withstand pressures more than 1,000 times greater than atmospheric pressure at sea level. Pressures found only in the very deepest ocean trenches, in the fluid inclusions of deep rock, and in environments that have no analogue in any human experience. And they do so without the complex pressure adaptation machinery found in deep sea animals, using only the ancient and elegant chemistry of a single cell. Piezophiles, as pressure-loving bacteria are called, have adapted their cell membranes to remain fluid and functional under conditions that would compress and immobilize the membranes of most bacteria. The fluidity of a membrane, its ability to flex and move, is essential to the function of the proteins embedded within it. Maintain that fluidity at extreme pressure, and the cell can continue to function. The membrane of a piezophile is, in this way, a kind of quiet miracle, a biological solution to a physical problem worked out over evolutionary time in the deepest and most silent places on Earth. Scientists have discovered bacteria living in the fluid of healthy human eyes, a finding that challenges the long-standing medical assumption that the interior of the eye is a sterile environment sealed off from the microbial world by the tight junctions of the blood-eye barrier, and suggests that the microbiome may extend into regions of the body that were previously thought to be beyond its reach. The bacteria found in ocular fluid are present in small numbers, and their role, whether beneficial, neutral, or potentially relevant to certain eye conditions, is an area of active research. But their presence there, quiet and unexpected, is a reminder that our understanding of the microbiome is still unfolding, still being revised by discoveries that find life in places we were certain it was absent. The body is not a sealed space with microbial territories clearly marked. It is a continuous gradient of life from the skin inward, more inhabited than any map we have yet been able to draw. NASA scientists have detected organic molecules and chemical signatures in meteorites found on Earth that are consistent with the byproducts of biological activity, compounds that, on Earth, are produced by living things, and whose presence in ancient space rock raises questions that science has not yet fully answered about the relationship between chemistry and life in the broader universe. These meteorites, some of which are billions of years old, carry within them a chemical record of the environments from which they came, the asteroids and planetary surfaces and clouds of molecular gas that make up the interstellar medium. Finding the molecular precursors of life in these ancient rocks does not prove that life has existed elsewhere. But it suggests that the chemistry of life, the amino acids, the carbon compounds, the molecular building blocks, may be not a rarity in the universe, but a commonplace, something that happens wherever the right conditions permit, on a cosmic scale that we have barely begun to explore. The panspermia hypothesis, the idea that bacteria or bacterial spores sealed inside meteorites could theoretically travel between planets and seed life across the universe, is taken seriously by a significant number of scientists as a genuine possibility, though it remains unproven and deeply debated. The physics is not the obstacle. Rocks are ejected from planetary surfaces regularly by meteor impacts and can travel between planets on timescales of millions of years. Some bacteria can form spores that survive the cold and radiation of space for extended periods. The question is whether any organism could survive the full journey, the ejection, the transit, the heating of atmospheric entry. No one knows. But the question is not absurd, and the possibility that life on Earth may have arrived in some deep and distant sense from elsewhere, or that life from Earth may have seeded somewhere else, is one that science holds open with genuine curiosity, like a door that has not yet been opened. Bacteria were among the very first living organisms deliberately sent to space, launched aboard early American and Soviet rockets in the 1950s and 1960s, in experiments designed to study how living things respond to the conditions of spaceflight, the cosmic radiation, the microgravity, the vacuum, the thermal cycling between the cold of shadow and the heat of direct sunlight. These early experiments were, in a sense, the first steps of astrobiology, the science that asks where life can exist and what it can endure. Bacteria were the obvious choice: small, easy to culture, genetically well understood, and already known to be remarkably resilient. Some of those early experiments returned results that surprised the scientists who conducted them. Bacteria survived in space or near space better than expected. The data began to accumulate that life, at its most elemental, might not be as fragile as the conditions of space suggest. Experiments conducted aboard the International Space Station have shown that some bacteria become more virulent, more capable of causing infection in microgravity conditions, producing changes in gene expression and biofilm formation that researchers are actively studying in order to understand the implications for human health during long-duration spaceflight. The microgravity environment of orbit changes the behavior of living things in subtle and sometimes unexpected ways. For bacteria, the absence of gravity appears to alter the fluid dynamics around the cell, changing how nutrients and signals are transported in and out, and triggering genetic responses that make some species more aggressive. The bacteria are not trying to harm astronauts. They are simply responding in the way that all living things respond to a change in their environment. Understanding that response is one of the many biological challenges that stand between us and the long-duration space missions that the future of human exploration may require. Tardigrades, the microscopic animals sometimes called water bears, famous for their ability to survive in space, in extreme cold and heat, and in conditions of total desiccation, share some of their extraordinary radiation survival genes with bacterial ancestors, in a case of horizontal gene transfer so extensive that roughly 1/6 of the tardigrade genome appears to have been borrowed from bacteria, fungi, and other microorganisms over the course of evolutionary history. This discovery, made when the tardigrade genome was first fully sequenced, astonished geneticists. It suggested that the tardigrade's legendary resilience is not entirely its own invention, that it has, over evolutionary time, helped itself to the survival toolkit of bacteria, incorporating bacterial genes into its own genome and making their ancient chemistry its own. The line between the bacterial world and the animal world is, in this case, drawn with a much softer hand than we usually imagine. Scientists searching for evidence of past or present life on Mars look specifically for biosignatures, chemical traces that bacteria are known to leave behind in the rocks and soils and atmosphere of the environments where they live. Isotopic ratios altered by metabolic activity, organic compounds associated with biological processes, mineral formations that bacteria are known to produce. Mars once had liquid water on its surface. It once had a thicker atmosphere. The conditions that prevailed on early Mars may have been similar enough to early Earth that if life arose on Earth in those conditions, it might have arisen on Mars too. The bacteria we study in Earth's most extreme environments, in hot springs, in acidic soils, in the deep rock, serve as models for what life on Mars might have looked like. We study Earth's bacteria, in part, to know what to look for when we look at another world. Enceladus, a small, ice-covered moon of Saturn whose surface is marked by enormous geysers that spray liquid water and organic compounds into space from an ocean that lies beneath the ice, is considered by many astrobiologists to be one of the most promising places in the solar system to search for microbial life because the conditions in its subsurface ocean closely resemble those of the deep-sea hydrothermal vent environments on Earth where bacterial life thrives. The Cassini spacecraft, which spent 13 years studying Saturn and its moons before its mission ended in 2017, flew through the plumes of Enceladus and detected water vapor, ice particles, carbon dioxide, methane and molecular hydrogen, compounds that, on Earth, would be consistent with the kind of chemistry that supports chemosynthetic bacteria. The ocean of Enceladus has never been seen, but the plumes it sends into space are speaking to us, very quietly in the language of chemistry, and scientists are listening with great care. The conditions inside certain deep-sea hydrothermal vent systems on Earth, the black smokers and white smokers of the mid-ocean ridges, where superheated water rich in minerals vents from the ocean floor into the cold deep sea, so closely resemble what scientists believe may exist in the subsurface oceans of icy moons like Enceladus and Europa, that researchers use them as direct models for what extraterrestrial microbial life might look like and how it might function. The bacteria that live at hydrothermal vents on Earth do not need sunlight. They do not need a surface. They do not need any of the conditions that we might instinctively associate with habitable environments. They need heat and chemical gradients and liquid water. If those conditions exist beneath the ice of an ocean moon, as the evidence increasingly suggests they might, then the possibility of life there is not a fantasy. It is a hypothesis, held carefully and tested patiently by scientists who have already found life in the strangest places on the world they know. If bacterial life exists elsewhere in the universe, scientists believe it would most likely resemble the extremophile bacteria found in Earth's harshest and most inhospitable environments, the heat lovers and cold-tolerant species, the acid dwellers and radiation resistors, because those are the organisms that demonstrate how far from our comfortable assumptions about habitability life is capable of reaching. The universe is not, in general, a gentle place. Most of space is cold and airless and dark, punctuated by the fierce radiation of stars and the occasional violence of collision and explosion. Life, if it exists beyond Earth, would need to have found its own relationship with those conditions, the way extremophile bacteria have found their relationship with the hot spring, with the reactor pool, with the deep rock. The bacteria in the most extreme environments on Earth are not curiosities. They are, perhaps, our best existing models for what life in the wider universe might look like. Some scientists believe that bacteria, in one form or another, in one extreme environment or another, on one world or another across the vast and populated reaches of the universe, may be the most common form of life that has ever existed anywhere, and that if the cosmos is inhabited, it is inhabited primarily, perhaps overwhelmingly, by the smallest and the oldest and the simplest kind of life that we know. Bacterial life arose on Earth within the first billion years of the planet's existence. It has survived every catastrophe. It has colonized every available environment. It has persisted unchanged in its essential form across more than 3 billion years of geological and biological upheaval. If life follows the same tendencies elsewhere in the universe that it has followed here, if it emerges early and persists through adversity and finds a way into every available corner of the physical world, then the universe may be quietly, invisibly, patiently alive with something very much like what lives in your body and beneath your feet, and in the warm soil of every garden that has ever been tended on a pale blue world drifting through the dark. And so we rest here, at the edge of everything we have traveled through tonight. A world older than imagination, a life smaller than a whisper, a presence so vast it fills the oceans and the sky and the deep rock and the quiet interior of your own body, working patiently and without pause, as it has always worked, since long before there were eyes to see or minds to wonder. The bacteria do not know we are grateful. They do not know we have been watching and learning and finding them beautiful in their ancient and invisible way. But they are here, as they have always been here, in the soil and the sea and the air and the warm places inside you where your own biology is endlessly, quietly tended. Let that be the last thing you carry into sleep tonight. That you are never alone, that you are cradled in life, that the world beneath the world is full of something very small and very old and, in its own patient way, full of wonder.